Hi guys, Miss Francis here to talk to you about alterations of chromosome number or structure that can cause genetic disorders. Some of these will be a repeat of things that we discussed during our mitosis-meiosis unit, such as non-disjunction. So non-disjunction occurs when there are problems with the meiotic spindle, and then that causes an error in the daughter cell because the chromosomes fail to separate properly. For example, during meiosis 1, during anaphase 1, if the tetrads don't separate properly, I have non-disjunction. Furthermore, during anaphase 2 of meiosis 2, if now my sister chromatids fail to separate, that's another example of non-disjunction. So if this non-disjunction occurs during meiosis 1, I end up with 4 gametes that all have incorrect numbers of chromosomes. These 2 gametes have 1 less chromosome, and these 2 gametes have an additional chromosome. However, if non-disjunction occurs during meiosis 2, I have 2 gametes with the normal number of chromosomes, but I have 1 gamete that's got an extra chromosome and 1 gamete that has 1 less chromosome. Now, what this non-disjunction leads to is different potential chromosomal disorders, such as trisomy. So trisomy is when the cell has 3 copies of a particular chromosome. The most well-known example of a disorder that occurs because of trisomy is Down syndrome, where you have 3 copies of chromosome 21. So if you were to look at a karyotype of someone with trisomy, do you see how you have 1, 2, 3 chromosomes at the 21st location instead of 2? That's trisomy 21, also known as Down syndrome. Other disorders related to trisomy include Klinefelter's. Klinefelter's is when a male has XXY sex chromosomes. These individuals have male sex organs, but they're sterile. They have feminine characteristics, but their intelligence is normal. Some other disorders related to trisomy is trisomy X. So a female is XXX. It occurs once in every 2,000 live births, but it produces a healthy female, so we don't often hear about that. On the other hand, monosomy or Turner syndrome is when a female has XO. This female looks normal. Her appearance is normal. So in other words, she's phenotypic. She's typical, but she is sterile and she is often immature. Polyploidy you don't need to know about, but if you're interested, plants are organisms that are usually polyploidy. They contain 2 complete sets of chromosomes. There are some alterations, such as breakage, that occurs in a chromosome that can lead to 4 different types of changes in chromosome structure. So now we're talking about mutations. So deletion occurs when part of a chromosome is missing. It's deleted. For example, Cri-du-chat, which is the deletion of chromosome number 5. Duplication happens when a fragment of a chromosome is duplicated. For example, Huntington's, and let me show you what that might look like. So, for example, deletion removes a segment, so segment D is removed. Duplication repeats a segment, so now I have 2 BC segments, deletion and duplication. Then we've got inversion and translocation. Inversion is when sections of a chromosome are inverted, so they read backwards to forwards. And translocation is when a chromosomal fragment joins a non-homologous chromosome. And let me show you what that might look like. So in inversion, instead of reading A, B, C, D, E, as this section of the chromosome is supposed to read, it's inverted, it's backwards, and now it reads A, D, C, B. Translocation moves a segment from one chromosome to another chromosome, one that is non-homologous. So here are my non-homologous pairs of chromosomes that have accidentally exchanged chromosomes. Deletions and duplications are common in meiosis. Duplications and translocations are the mutations that are typically harmful. Cri-du-chat, we talked about, it's deletion in chromosome number 5, and unfortunately it is fatal. Some of these mutations can be caused by environmental factors such as radiation. Radiation alters your DNA, and if your DNA is altered, then it may result in a disease or a disorder. Mitochondrial DNA has genes too. Mitochondrial DNA comes from your mom, and usually the disorders that are associated with mutations in mitochondrial DNA are very rare, and they primarily impact ATP supply by producing defects in the ETC or ATP synthase. So therefore, the tissues that require high energy supplies are affected. For example, the nervous system, muscles, and they may suffer energy deprivation. Other mitochondrial mutations may contribute to diabetes, heart disease, and other diseases of aging. This is our final video on the topic of Mendelian genetics. If you have any questions, please come see me for extra help.